Okay, this lecture is on the structure and function of the cell. So we are going to focus on basic definitions. We're going to first cover some scientists, and then we'll get into the parts or components of a cell and look at every function. And that's really going to be the focus of this particular objective. So what is a cell? It is the smallest unit, and it can carry on all the processes of life. So if you think of it as how your body works, it also works at the cellular level. Okay, now there are some scientists that we need to be familiar with. The first one is Robert Hooke. And don't worry about the dates. I'm not going to ask any dates on the exam. Just to give you a reference and time of when this took place. So Robert Hooke, he worked a lot with the microscopes, very primitive microscope. And what he saw when he looked in the microscope, let me pull up his picture here. There's his microscope. So these are the actual images of the cork cells. So he looked at dead plant cells. That's very important. There's another scientist that looked at living cells. So what he did, cork is a dead plant, so he would make a cross-section of the cork, different types of cork, different kinds of wood, and when he looked into his microscope, which is a scope right here, he saw little boxes, and all the little boxes reminded him of the rooms in the monastery, and they called those cells at that time. I think it's kind of interesting that they called them cells, and now prisoners live in cells. So, and I'm not sure if the monks still refer to them as cells. Okay, our next scientist, kind of a mouthful, Anton von Leeuwenhoek. I've heard it pronounced a couple of different ways. But he made microscopes, and his time frame was about 10 years later after Hooke, and he focused on observing different types of living cells. So that's really what he focused on. Now there are three other scientists, and together collectively with all their observations and theories, they came up with what we use as a universal cell theory. This is very basic. There's actually 7, even 8 parts to the cell theory. But we're just going to look at 3 main components of the cell theory. First is Schleiden, and he was a botanist, so we know that he's focused on plants, and this is him right down here. And then there was also Schwann, and he focused on zoology, so animals. And collectively, the two of them concluded that all living things are made up of or composed of cells. And this is Schwann. I have to throw a little funny in there. So every time I see this scientist, I always think of ice cream, the Schwann truck, but that's not the case. Okay, our third scientist is Virchow. And here's a picture of him. And he proposed or came to the conclusion that all cells come from pre-existing cells. But now we know that that is called mitosis. So cells make other cells. So together they came up with, or they are given credit for the cell theory. So the 2 main parts was, the first one was kind of reworded, and this came from Schleiden and Schwann. Cells are units of structure and function. So that's going to be our next part that we're really going to focus on, the parts inside the cell and what do they do to make the cell a living structure. And then Virchow, all cells come from pre-existing cells, or mitosis. Okay, so we looked at 5 different scientists. We're going to kind of make a transition on shape of cells, and then we'll get into the parts inside the cell. So cells can take on many different shapes and form, and it kind of depends on their function. So they're very closely related. So cells could be cubed or squared in shape, and usually when they have that structure, they're more related to the plants, or they can be round or spherical, and we associate that more with the animal cells. Now, I had said that it depends on where you find these cells. So if I have a very flat, irregular shaped cell, it would be used more for a covering. So that would be like our epithelial cells or our skin cells, flat, thin, irregular shape, kind of fit together like a puzzle, and so they're used for protection and covering. Okay, so now let's get into the inside parts of a cell. And I'm also going to point out some things that how plant and animal cells differ from one another, and you really need to pay close attention to that. So an organelle. So I'm going to be referring to all the parts or components inside the cell as organelles. So you can think of it, when we went back to chemistry, that the atom would be like the cell, and the parts inside the atom were the subatomic particles. Well, in the cell, they are the organelles. So if that helps you remember that, then that'd be good. Okay, so what is an organelle? It literally means a little organ. So a cell is just like our living inside our body. So our body is made up of cells, and in each little cell, it has its own functioning unit. Okay, so let's look at the different parts. So we're going to do a little review here. We have our eukaryotes and our prokaryotes. And in the last chapter, we talked about what kind of organisms we find in these two general classifications. And remember I said that you are a eukaryote. So those are our higher level organisms, plants, animals, fungi. But the key difference between a eukaryote and a prokaryote has to deal with the nucleus. So a eukaryote, plants and animals, they have a nucleus. And the other thing is the membrane around the organelles. So we're going to see membranes definitely around the cell itself, around the nucleus. We'll also see membranes around the mitochondria and the chloroplast. So those all have membrane-bound organelles, is what that means. Prokaryotes, they lack a true nuclei. They're still going to have the membrane-bound organelles, but the nuclei is very loose. It's called a nucleoid. We're not going to get into the parts of a bacteria. We're really going to focus on plants and animals. But the reason why it's not compact and in a nucleus like the eukaryotes is so it can replicate very quickly. So you know, if you get a cut on your finger, and it can get infected very quickly, and it's because the bacteria can replicate so quickly. Okay, so the parts of a cell. This part will be on the test as a multiple choice. So the three main parts of a eukaryote. First is a cell membrane. Okay, both plants and animals have a cell membrane. Cytoplasm and a nucleus. Okay, so three main parts. So let's break down the three parts, and we're going to also add some other organelles to that you'll need to be familiar with. So the cell membrane is also called the plasma membrane, and I don't care which one you use. I may use those terms interchangeably. And for the animal cell, that is the only membrane. That's it. Okay. For a plant cell, it has the innermost membrane, the cell membrane, but then it also has a thicker wall called the cell wall. And I'll get to this in just a second, but the reason why is so the plant needs structure. There's no skeletal system like for animal cells, so it needs to have that thicker cell wall. We've kind of been talking about with osmosis and diffusion, selectively permeable in our lab that we have done on diffusion, things moving in and out. So it's these membranes that allow things to pass in and out of the cell. Okay, phospholipid layers. That just describes how the membrane is composed, and I'll pull up an image here in just a second. But we'll see protein embedded, carbohydrates. So those are our macromolecules. We're going to see that throughout the year. Okay, here's our cell wall. Now this is one thing that I will pull out. You'll see it on your study guide and a couple other worksheets. Cell wall. Cell walls are only in plants, not animals. So that'd be a really good question on the test. And I'd already stated why: support and to protect the cell. And keep in mind, just to the inside of that is a cell membrane, whereas the animal cell membrane, that's it. Okay, so here's our cell membrane, and here is our lipid layer that I was talking about. So there's lipids, one of our macromolecule. So a lipid we know is kind of fatty. So we see here's a term, and I'm not going to ask questions about this, but to kind of explain why it looks the way it does, We have what's called a hydrophilic. That means it likes water. Hydrophobic, kind of like you're hydrophobic about something, it does not like water. So you notice these tails are pointing in toward each other. And what it does is when things pass through the membrane, it's literally like a magnet system. It pushes and pulls. So it likes it and then it repels it. So again, I'm not going to ask that, and that's a very crude explanation of that. But together, this is a cholesterol, and lipids, cholesterols work together. So I can pull out and see the different macromolecules. So here's a carbohydrate, here's a lipid, and these are different proteins. And what's not shown in here in this cross section are gated channels, which we talked about earlier. Okay, then another thing we need to be familiar with is called the cytoplasm. Now, if a cell does not have cytoplasm, it's not living. It cannot reproduce, it cannot replicate itself, it's dead. So the cytoplasm is, and it's kind of hard to see, but these pink dark outlines, these are cells, and these are animal cells because they're kind of rounded, no square edges. So here's another one. And the dark pink structure inside is the nucleus. We'll get to that in a second. And then the darker pink is the nucleolus. So all of this lightish white pink inside filling this is the cytoplasm. And the definition is a jelly-like material. It has water and some salts. And what it does is constantly moving. Think of it like Jell-O. I don't know if your mom or your grandma has ever made you Jell-O, and they may have put fruit cocktail in it. And so all the little pieces of fruit floating in the Jell-O, think of that as your organelles in your cell. So the bowl is the cell, the Jell-O is the cytoplasm, and the little floating pieces of whatever it is, fruit, are the different organelles. So hopefully that's a way to help you remember that. Okay, here's the nucleus. This is whole tannish structure. And this red ball in the middle is the nucleolus. Now I do want to point out, and I'll say it again a little bit later, never learn or memorize a structure and what it looks like based on its color. It will fail on you every single time because depending on where you get the image, who draws it, what computer generated it, anyone can make it any color. So do not rely on color. Look at the structure and the components inside the organelles. And we've got a few that look a lot alike, but I'm going to point out how you can tell them apart. Okay, so this nucleus has, and it's kind of hard to see, these yellow structures in this case, has all the DNA and the chromosomes. The nucleolus is the ball inside, and that is responsible for making RNA. So DNA, RNA, another macromolecule. Now, nucleolus, nucleus. This is the case where spelling will count. No one letter off because they are so close. So you're just going to have to memorize these, and you're just being told now. Spelling counts definitely on these two. So the job of the nucleus, and I'll try to give you ways to help remember it. The nucleus is like the brain. It controls everything that happens in the cell. So we've already talked about nucleic acids, DNA. I could also add RNA. Nucleolus is associated with making ribosomes, RNA. They'll go together. We'll see how that really happens next semester. Okay. And then surrounding it, here's another membrane. I had mentioned it earlier. This membrane, you can also call it nuclear envelope. I learned it when I was going through school, nuclear membrane. It just makes more sense. But it's kind of hard to see, but it's a sandwich just like on the cell membrane. So make sure if you're doing any worksheet, lab, or the test, you be very clear to indicate nuclear membrane or cell membrane. They are two totally completely different things. Okay, ribosomes we had just mentioned are associated with the nucleolus. So the ribosomes also has its own function. So the nucleolus helps make ribosomes, and then once the ribosomes are made, They are in turn are going to make protein, and we know that that is another macromolecule. And protein is found throughout the cell. It's used, we've seen it in the cell membrane. It's going to be used for various structures. Very, very important macromolecule. Okay, so here is a drawing of a ribosome. Now, this is a very close-up of a ribosome. When you usually see it in the cell, and I'll point it out on another structure, they look like little dots, like seeds. But, and I'll say this again, we'll examine this a little more next semester, but it's actually two pieces that fit together called a subunits. And what happens is in between these two units, because you got this dark purple, light purple, together they make a ribosome. And I'm not going to ask any questions about this on the test, but a special molecule goes and fits right through here, kind of snakes through there, and when it goes through there, that's how it makes the protein. So for right now, all you need to know is that the ribosomes make protein. Okay, now this one, even though you may see it abbreviated, I do want you to know how to spell it out. The endoplasmic reticulum. Okay, so what that is, and let me pull up a picture of it. Okay, this is a 3D example, and here is the nucleus. So this darker purple. See, the other one was tan, now this one's purple. Okay. They're all folded sacs, okay? And if you think of it, and this is, again, how I learned it, it's kind of like a highway system or streets. You connect homes, you connect towns. So the ER connects organelles. Okay, so maybe the nucleus is like the capital, and maybe it's connected over here to a house. Okay, that's just an example. Okay, there are two types of ER, endoplasmic reticulum. There's one that's smooth, and there's one that's rough. And the difference between the two that we're going to focus on at this level is that the rough have the ribosomes or the protein, and you can see they look like little dots on the darker pink, and the smooth, they have no proteins or ribosomes. So that's all we're going to concern ourselves with. Actually, they do distinguish in as far as what they make or synthesize, but just being able to tell them and distinguish them and know that they are tubes and channels that connect organelles. Now let me pull up, here's another example of color not being very reliable. So here's the rough, you can see the dark dots, okay, those are all the ribosomes. So see here, it's labeled ribosomes. Some, it may be ribosomes protein or proteins, okay, so sometimes they do use those words interchangeably, but knowing that the ribosomes make the protein, sometimes they'll just say it's covered with protein. So let's just say it's covered with ribosomes, okay? So this one, in this case, is green, and this one is dark pink. The smooth ER, this one's kind of peach color, and this one's kind of tannish green. But they still are tubes, and they're interconnecting channels. Okay, this is the one that looks a lot like the ER, Golgi apparatus. I've also seen it in books where it's called Golgi body, spelled B-O-D-Y and B-O-D-I. I'm familiar with Golgi apparatus. That's probably the term I'm going to use. And recently, you know how botanists are, they now have their own term, and this is just in the last few years. They call the same structure a dictosome. So let's just call it Golgi apparatus. But if you put body, that's fine. What it does, they are stacks of membranes or sacs. And so this is kind of, this first top view, it's looking down on it, and what they've done is they sliced it open and kind of tilted it towards you. So they can be kind of flat sacs, kind of elongated, they can be kind of smaller sacs. Okay, so this is looking inside. The job or the function is to process, package, and secrete. So to me, it reminds me a lot like a post office. So what do? You gather all your mail, you send it to the post office, they process it by putting it by the zip code, Then they're going to package it up, put it in the mail truck, and then the mailman's going to deliver it or secrete it. So hopefully that'll help you remember this. So this is where, and here's the protein again, so this is where the protein goes to kind of do the final assembly. Okay, here's another view, kind of, they've cut it across now, tan versus purple, but here's what you need to look for, and I've got another image to put them side by side between the Golgi and the ER. The Golgi apparatus, you can follow the lines and see they are individual stacks, individual sacks. They're not connected. So think of it also, if you want to, as a stack of pancakes. Each pancake is a flattened sack. Okay? So putting them side by side, here's the ER, the pink, and if I follow the grayish sacks, I can see it's connected to the one below it. This one's connected, and here's where it shows, remember the ER connects organelles? So here, a little vesicle with something in it is going to transport it to the Golgi, and the Golgi now is going to bring that in, process it, package it, and then once it's done doing that, you can see here, now it secretes it, and now it's leaving the cell, because here's the plasma membrane. So it's a secreting vesicle. And this one is kind of a lighter tan, but they are actually, sorry, that was my dog. This isn't actually, those are not connected like up here. So you need to look inside to see if it's an ER or a Golgi. Okay, mitochondria. Now the mitochondria is, they call it the powerhouse of the cell, but its job is to make energy. Energy for the cell to carry on all the processes, anything, moving things from one place to another, synthesizing something, whatever the case may be, is the energy, the food. The name of the energy that is formed is called ATP. We've seen that before. So when they say respiration, it's not that the cell is breathing in and out like we are. It's cellular respiration. I probably should have wrote the word cellular. But it's going to do a lot of converting, go through metabolism. We'll look at cellular respiration, photosynthesis next semester. But for now, just know that the mitochondria makes energy, and the name of the energy is ATP. Now, this is another test question. I will have on there what two organelles have its own DNA. One, it says it right here, is the mitochondria, and the second, I hope that you would say, is the nucleus. Now we know why the nucleus has its own DNA. Obvious reason, so it can replicate and make new cells. But why would the mitochondria have its own DNA? No other organelle does. The reason why is if that cell needs energy, it needs it immediately. It cannot wait for the nucleus to go through the process to make more mitochondria, more DNA for itself. So what it's going to do, if it needs energy now, it can go ahead and replicate or make more mitochondria as the cell needed. For example, if you are hungry and you need food, you go to the refrigerator, you take care of it yourself. Okay, so here's the mitochondria, and just like we saw in the Golgi and the ER, they're very similar in structure. This organelle and another organelle look the same, so we got to look inside. This is a very classic image of a mitochondria. This is a very not typical image of a mitochondria, but they're both the same. Again, this one's computer generated. The artist decided to pick some kind of neon colors, and that's fine. So how can I tell them apart? How do I know that's the mitochondria? Looking inside, I can see that this looks like a ribbon going back and forth. These are membranes. Inside the membrane, it has electrons, and that's how it makes its own energy to make ATP. Look inside here, here's the ribbon going back and forth, back and forth. Okay, so remember the ribbon inside. I always thought this looked like a slipper. This very typical, and you can tie up your slipper with a ribbon. Okay, a lysosome. Now lysosomes are kind of interesting little organelles. The fact that they have digestive enzymes inside it. So when I have an old organelle that's kind of wearing out, it will send it to the lysosome and it will break it down. Similar to our stomachs, okay, any old parts. Now what's kind of cool is that inside, when the embryo is developing, inside the womb, when we're developing, I'm going to hold my hands up here, we start off with, we have something called the limb buds, okay, both our feet and hands. The tissue is connected, so we're not able to open and separate our fingers in the early stages of development. These lysosomes, a gene turns on the lysosomes, and it eats through the digestive enzymes the tissue away to form our fingers and our toes. If that gene turns off too quickly, it could leave a little webbing in between your fingers and toes. So there's a little fun fact for you. So here's a lysosome. Again, they're not all green, but this one happens to be green. Now, how can I tell this organelle from another organelle that looks exactly like it if it wasn't for the color? Well, since it has enzymes in it, it always has this appearance of kind of a grainy, gritty look to it. Okay, so it looks like it's kind of swirling and there's something digesting inside it, so very granular. So look inside it again. Okay, this one looks a lot like the lysosome. This is called a vacuole. Same shape, maybe a little more oval, but you notice there's no granular swirling business going on. Very clear, fluid, and the reason why is filled with mostly water. Now, you mainly find this in plants. In a plant, a vacuole will take up about 90% of the cell. Quite a bit of it. Everything else is pushed out of the way. So when we talked about plasmolysis, cytolysis, focusing on water going in and out, the reason why is because 90% of it is in this organelle. So whatever the water is doing, the cell is reacting to it because 90% of that cell is made up of water. Okay? If it's found in an animal cell, it's very small, about the size of a lysosome. So that's why you need to look inside. Is it granular or is it clear and fluid? Okay, cytoskeleton. Now obviously cells at this level does not have its own skeletal system. So there's got to be something besides the turgor pressure and the vacuoles that's going to keep the cell expanded. So inside the cell, and you don't see it with the kind of scopes we have, there's something called cytoskeleton. Cyto, remember, means cell, so it's a skeleton for the cell. And here's protein again. See, protein's used everywhere. So protein make up these two different sizes of tubes that kind of cross, mishmash. They form X's and long tubes. So here's an image of it. So this is kind of looking at the cell. So the top part is one cell membrane. This is the bottom cell membrane. And you have these tan tubes, not the big blue ones, that's something else. But these tan tubes are the microtubules, okay, protein tubes. All the little X's that are connected by these black dots, those are threads, and those are called microfilaments. So I don't know if you ever looked up in the attic inside your house, but inside they have rafters, and the reason why they form these X's is it provides more support and stability if they are crossed as opposed to just going straight lines. Okay, we have cilia. Now this is on the outside of the cell, and so these are very short, and they're also made of protein, and you usually find these attached to cells that would move fluid or maybe have mucus associated with it. So in our respiratory system, on our cells, we would have the cilia that's kind of moving, and dust and dirt gets trapped in that. So that's just another little extension feature. Another that you might find on the outside of a cell is a flagella. It's longer than a cilia, It can occur by itself or in pairs, and we will primarily see it associated with sperm, and we'll look at that next semester. Again, made of protein tubes. Just a couple of images there. Okay, chloroplast. This one looks a lot like the mitochondria. Again, looking inside. Now, this one, most of them are green. The reason why is because they are associated with plants and the green pigment. So chloroplast, I hope by now you can figure out, is associated with photosynthesis, and only plants have photosynthesis. Pardon the phone. Okay, inside, it kind of looks like a green jelly bean. Inside are these stacks. Okay, so if you need to pause it to look at this, go ahead. But if you think of a stack of quarters, all of these are stacks, and then in between it, they're kind of connected, kind of this fluid. I'm not going to say a ribbon, okay, but this fluid is connecting these stacks. Together, they carry out photosynthesis, okay? Now, don't get chloroplast and chlorophyll confused, okay? Chloro means green, okay, so that's the green pigment. And it's the pigment that absorbs the sunlight that carries out photosynthesis. Plast, P-L-A-S-T, is the actual structure. So this is the plast, the chloroplast. Phyll is pigment. And I had a professor who said one time, Phil is a pig. And it just kind of stuck. Okay? Another structure are the centrioles, and we mainly associate this with animal cells. Plant cells have a little bit different structure, and sometimes a teacher will say, okay, centrioles are in both for mitosis, etc., but primarily in animal cells. So if we needed to distinguish between plant and animal cells, we will put centrioles with the animal cells. Okay? And it's used for cell reproduction, more specifically mitosis. So these guys are going to hang out near the nucleus, and that's their only job, is to help separate the nucleus. Okay? Okay, so that was kind of the cell structure and function. And the last little tidbit, it's a very small part in your book, multicellular organization. I'm not going to spend a lot of time on this. Cell specialization, the definition is division of labor among cell types. All that means is that each cell is designed in the structure to carry out a particular function. Okay? So if I have a very flat cell, like at the beginning, flat, irregular, covering, its job is going to be used for covering skin, etc. Okay? It's not going to then suddenly become a heart cell. They all have their own function and develop accordingly. Okay, now this kind of looks like our food chains that we did, but I drew it this way so you can see how they all build on one another. So the organelles make up a cell. Cells that function together, notice I use the word function, will make up a very specific type of tissue. Tissues that function together will make up an organ. So for example, I may have 3 different tissues, and together they're very similar. They would make up the heart. Okay? And then I would have organs, heart, veins, arteries, and that would make up a system, or the circulatory system. Okay? Or the cardiovascular system, etc., whichever system you want to say. Now I threw skin on here because it's kind of tricky. Skin can be classified by its function as either a tissue or an organ, and it's just because it's an epithelial cell. Okay, so I'm not going to try to trick you on that. So if I did put that on there and you put tissue and someone else put organ, you would both be correct. Okay, and I gave you a couple other little examples. Okay, last thing here, a colonial organism. You may read this in the book. You have some organisms that kind of jump from going to a cell, and then when they come together, they don't form tissues, they don't form the organs or the systems. They make up a complete organism. Okay, so like if I draw another arrow, systems, when I get all the systems together, the 12 systems of a human, then I make a human. So, but I got to have 12 systems. Whereas here, something like green algae, if I have a group of cells together, let me pull the picture up here, all these little dots together, those are cells, and when they all come together, they're going to make an actual organism. Okay, so there's not tissues there, there's no organs, there's no individual systems. It simply goes to an organism. Hopefully I didn't kill that. Okay, so green algae, the genus name is Volvox, and so that's, you'll see that in a lot of textbooks. Okay, so one little final question here, going back to our scientists. Robert Hooke came up with the word cell. Okay, we know that's from his little rooms. What organelle or structure of the cell was he observing? Give it a little time. Dun, 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 dun. Okay, enough of my singing. He observed the cell wall. Remember, he observed cork, plant cells, okay, the cell wall, so he was looking at the outside membrane, hence cell wall. Okay, so hopefully that wasn't too long. Make sure you do know, there are a lot of questions on the test about the different organelles, their functions, and also being able to identify what they look like. Okay, so I will see you next time.